Hello friends, so in this video lecture we will discuss about the amino acids. Basically proteins are polymers of amino acids and amino acids are building blocks of proteins. An amino acid is an organic molecule that is made up of a basic amino group NH2 or NH3 and an acidic carboxylic group that is COH and an organic R group which is also called a side chain. An amino acid structure is an general structure of an amino acid that you can see on your screens. Amino acids differ from each other in their side chains or R groups, which vary in structure, size, and electric charges. The R group is basically a variable group, and the R group determines polarity, acidic and basic nature, that is pH, and also isoelectric points and molecular mass or size of each type of amino acids. Therefore, R group or side chain of an amino acid exert a profound effect on the structure and biological activity of proteins. Amino acids are classified by their R groups and the group's interaction with water. First is hydrophobic. The majority of R groups are hydrophobic in nature, and second is polar uncharged amino acids, and third is polar charged. First we discuss hydrophobic R groups. R groups composed of only hydrogen and carbon compounds, and one exception of methionine that contains sulfur, bound only to carbon compound. Second is your polar uncharged and charged amino acids. Look for oxygens and nitrogens. Charged groups will be charged or have a primary amine or carboxylic acid part. Primary amines are written NH2 and carboxylic acids COH. The next thing is amino acid can form a polypeptides. Peptide linkages, carbon and nitrogen bonds. Different order equals and different peptides. Example include glycine, alanine and protein. Is not the same as proline, aniline and glycine. Number of possible combinations using each amino acid once. Number of amino acid and factorial. Example include tetrapeptide is 4 factorial. 4 factorials mean 4 multiplied by 3 multiplied by 2 into 1 is 24. If it is 3 factorial, the sum is 4 multiplied by 3 multiplied by 2 multiplied by 1. If it is 2 factorial, so we do 2 multiplied by 1. But the example of tetrapeptide is 4 factorial. That is 4 multiplied by 3 multiplied by 2 multiplied by 1. That is equals to 24 number of amino acid. Number of possible combinations using each amino acids as many times as we want. Number of amino acids and number in polypeptide. Example, 4 amino acids in a tetrapeptide. It's mean 4 are amino acids included in a tetrapeptide, and there is a 4 factorial also included. To know the formation of polypeptides, you must know the formation of peptide bonds. What are peptide bonds? Amino acids are joined together by the formation of a peptide bond, where the amino group of the one molecule reacts with the carboxyl group of the other. Peptide bond formation is an example of a condensation reaction. The reverse reaction, bond breakage, involve water is an example of hydrolytic cleavage or hydrolysis that can further form a peptide bond. 3 amino acids can be joined by 2 peptide bonds to form a tripeptide. When a few amino acids are joined in this fashion, the structure is called oligopeptide. When many amino acids are joined, the product is called polypeptide. So in this way, amino acids can form polypeptides. Here you can see the Zwitter ions where net charge is 0 on the molecule, and the carbon is always an alpha carbon. Next is isoelectric point, the pH at which the molecule carries no net charge. It is an acidic solution, cationic, and alkaline solution, anionic. In low pH, it acts as a cationic, and hydrogen molecule is released whenever a pH increases. It is converted into NH3 to NH2 at high pH. Next is pK value. It is the ability of an acid to donate a proton, the degree of dissociation, also known as pKa or acid dissociation constant. The pK values of a alpha carboxylic group is in the range of 2.2, and 9.4 is the range of pK value of an alpha amino group. Next is titration curve of glycine. On x-axis you can see OH negative equivalents, and in y-axis here is the pH scale, and pK1 is the pH at which 50% of molecules are in cation form and 50% are in Zwitter ion form, and pK2 on the top, that is 9.60 value corresponds, and pK2 is a pH at which 50% of molecules are in anion form and 50% are in Zwitter ion form. The value in the titration curve of glycine is 5.97, that is an isoelectric point. These all amino acids are non-polar amino acids, include glycine, alanine, valine, leucine, isoleucine, phenylalanine, and many more. Here you can see uncharged amino acids named serine, threonine, triosine, asparagine, glutamine, and cysteine. Basically, these amino acids have zero net charge at neutral pH, so that's why it is called uncharged amino acids.